Chapter 14, DNA Structure and Function. We now know that DNA contains the instructions for an organism's genetics. DNA stands for deoxyribonucleic acid. Our knowledge of DNA has grown so rapidly that DNA can be gathered from hair, blood, or saliva. This can help us solve crimes, find out who our baby's father is, tracing genealogy, archaeological research, and tracing disease outbreaks. One conspiracy theory of COVID-19 in that glorious year 2020 was that it was a man-made virus. However, DNA testing shows us that it is related to the same coronaviruses that caused Middle East Respiratory Syndrome, MERS, sudden acute respiratory syndrome, SARS, and many cold viruses. Also, by testing coronaviruses, we've been able to trace that SARS-CoV-2, the technical name of the coronavirus we see now, was in fact naturally derived from bats in China. Most humans have 23 pairs of chromosomes, one set from mom and one from dad. We also inherit mitochondrial DNA exclusively from our mother, and you can receive some genetic disorders from this. Our chromosomes have between 20 to 25,000 functional genes. A gene is a sequence of DNA that encodes for a functional product. For example, the darker your eyes, the more melanin that is found in the irises. Melanin is a protein that is encoded from genes dictating your eye color. Those with blue eyes have less melanin expression in their irises. While our current model of DNA stems from the 1950s, the discovery of nucleic acids, the monomer units of DNA, go back as far as the 1860s, when the man by the name of Friedrich Miescher isolated phosphate-rich chemicals from white blood cells, which he called nucleons, since they were isolated from the nuclei of those cells. We later found out that phosphates make up a large portion of DNA's molecular structure. In 1928, Frederick Griffith, wow, DNA really loved Freds, didn't they? Demonstrated bacterial transformation. And transformation happens when external DNA is taken up by an organism changing its anatomy and physiology. To show bacterial transformation, Griffith used Streptococcus pneumoniae, a bacterium that causes pneumonia. He had two strains in the lab, rough and smooth. These were so named by how they appeared on the growth plates he used to grow them. The rough strain does not cause disease, while the smooth strain does. The smooth strain has a protective capsule that allows it to evade a host immune system, thereby causing disease. So Griffith performed 4 tests on mice injected with this bacterium. Mouse number 1 is injected with the S strain, or smooth strain, and dies of pneumonia. Mouse number 2 is injected with the rough strain, the one that doesn't cause disease, and so therefore it survives. Mouse number 3 is injected with a dead smooth strain, that is, one that's been heated to kill off any of the protein's DNA found within that virus. So therefore the mouse is going to survive. So finally, he decided to take some heated S strain, which was dead, and some living R strain, which is not disease-causing, and he tried to see what happened. And come to find out, the mouse died. So what happened? While Griffith couldn't find the answer, he did develop the transforming principle, and that something was passed from the dead smooth bacteria to the live rough bacteria. The dead smooth strain's DNA was released from the damaged cells and uptaken by the rough strain. This allowed the rough strain to cause pneumonia. In 1944, a group of scientists repeated Griffith's experiments, but isolated the proteins and nucleic acids thought to cause pneumonia to form from the S strain of bacteria. They used special enzymes that degraded these molecules. Afterwards, the S strain was placed in the mice, but the mice survived, leading them to conclude that DNA was the transforming principle that caused the S strain of bacteria to be so deadly. Unfortunately, at the time, many scientists did not think DNA had much to do with the storage of biological information. It was thought that proteins did. So in 1952, Martha Chase and Alfred Hershey studied bacteriophages. So bacteriophages are viruses that exclusively infect bacteria. Due to the simple structure of a virus, which is just a piece of DNA or RNA surrounded by a protein coat, they observed that the bacteriophage injects its nucleic acids into its host. In Hershey and Chase's experiments, bacteria were infected with phage radiolabeled with either radioactive sulfur, which tends to target proteins, or radioactive phosphorus, which labels or targets DNA. They noticed that only the radioactive phosphorus entered the bacterial cells, indicating that DNA is the genetic material and not the proteins. In the end, the phage containing radioactive phosphorus multiplied inside the bacterium, whereas the radioactive sulfur phages did not, leading them to conclude that genetic information is carried on DNA and not proteins. Also, in 1952, Erwin Chargaff studied the nucleic bases adenine, thymine, guanine, and cytosine, and found that these bases were not found in equal numbers, and the relative concentrations of these four bases varied from species to species. Perhaps even more intriguing is that the number of adenines and thymines observed and guanines and cytosines observed were equal, no matter what species was being studied. Different species add equal amounts of purines, which is a class of chemicals including the bases adenine and guanine, which have a two-ring shape, and pyrimidines, a class of chemicals including bases thymine and cytosine, which have a single hexagonal ring. But the ratios of adenine and thymine pairs and guanine and cytosine pairs differed. Thus, these observations provided us with Chargaff's rules, which are the amount of adenine is equal to the amount of thymine, and therefore the amount of guanines is equal to that of the number of cytosines. Also, all species have different ratios of adenine to thymines compared to the guanines and cytosine. For example, one species might have 60% adenine-thymine pairs versus 40% guanine-cytosine pairs. Another species might have 51% adenine-thymine, 49% guanine-cytosine pairs. However, the numbers of adenines and thymines will be equal, and the number of guanines and cytosines are equal because they pair up. And so that's going to be the next topic. How is DNA built? DNA is made of nucleotides, which are made of a 5-carbon deoxyribose sugar, a phosphate group, and one of 4 nitrogenous bases, adenine, guanine, cytosine, or thymine. DNA is a double-stranded molecule with a sugar phosphate backbone made with a strong phosphodiester bond. The nitrogenous bases are held together by weak hydrogen bonds. A and T are held by 2 hydrogen bonds, and G and C are held by 3 hydrogen bonds. Let's look a little bit more closely at a single nucleotide. DNA contains a pentose sugar called deoxyribose. This configuration means that deoxyribose has 5 carbons, and we call the carbons 1', 2', 3', and so forth carbon. The 1' carbon is bonded to the nucleotide base. The 3' carbon is bonded to the phosphate of another nucleotide. It forms a phosphodiester bond by doing a condensation reaction with another nucleotide. The 5 carbon is bonded to the phosphate of the other nucleotide. One thing that's important to remember is these help us provide directionality. For example, we say that this strand of DNA is going from the 5' to 3', 5' to 3' direction. If we want to go the opposite way, we say 3' to 5', 3' to 5' direction. Kind of similar to saying north and south and east and west. So these are directionality terms that we'll be using to help us understand how DNA is replicated and how transcription and translation, which we'll talk about in the next chapter, occurs. So why does adenine and thymine and cytosine and guanine combine? Adenine and guanine are purine bases and are larger than cytosine and thymine, which are pyrimidine bases. Adenine and guanine are too large, and that if they were to pair together, it would end up crowding the space between the 2 DNA strands, preventing DNA from being accessed by the cell. Likewise, cytosine and thymine are too small, which would be too far apart to create a hydrogen bond to hold the 2 strands of DNA together. Also, adenine and thymine share 2 hydrogen bonds, and cytosine and guanine share 3 hydrogen bonds. So this helps to explain why A goes with T and G goes with C. All right, back to our history of DNA. So who discovered the final shape of DNA that we now know as a double helix? Working on the research of others, Rosalind Franklin used an X-ray diffractor to determine that DNA's shape was a double helix. Unfortunately, history overlooks her, and I can't seem to understand why. James Watson and Francis Crick built upon Franklin's research and was able to model the double helix into the amazing shape we see today. In 1962, Watson and Crick won the Nobel Prize in Medicine for their discovery of DNA shape. Unfortunately, Franklin had died and she had not received the award since Nobel Prizes are not given posthumously. Interesting fact, James Watson is still alive as of 2020 and is over 90 years old. It's funny when we look at these pictures as if they are found in a distant past and that these people are all gone. And so, therefore, this was the final model that was produced by Watson and Crick based on previous research. So, DNA sequencing technologies really took off in the 1990s once cost and safety was addressed, as well as greatly reducing the amount of time it takes to perform them. The first sequencing method is called the deoxy chain termination method. It uses chain terminators called deoxynucleotides, or DDNTPS. These differ from DNA in that the sugar lacks a free hydroxyl group on the 3' end, so this prevents a growing chain of DNA from continuing, allowing scientists to make certain fragments of a fixed size of DNA. Gel electrophoresis is a technique used to separate DNA fragments of different sizes. This uses a gel made of a chemical called agarose, which is heated to a liquid form and then placed in a casting tray to cool. DNA is then added to these wells made from a plastic comb added during casting. A liquid buffer solution is added to the electrophoresis chamber where an electric current is applied. DNA is negatively charged and is pulled to the positively charged end of the chamber. Larger fragments move slower than smaller fragments, thus separating these fragments. Once completed, the gel is dyed to make it easier to view the DNA fragments and it is then analyzed. The purpose of gel electrophoresis is to compare a known DNA sequence against several unknown and can be used in criminal investigations, paternity testing, and species identification. So, while prokaryotes have all of their DNA situated in a folded circular chromosome in a nucleoid region, regardless, pros and eukaryotes employ supercoiling techniques to wrap DNA around proteins used to compact lots of DNA into a tiny little space. Eukaryotes, which have a large linear strand that divides into separate chromosomes during cell division, use histone proteins to coil DNA around. DNA replication is a semi-conservative process, meaning that DNA is divided into two, and each new DNA strand produced contains a strand of the original DNA and a strand of new DNA. And up next, we're going to look at the processes of DNA replication in both prokaryotes and eukaryotes, as well as what happens when there's mistakes made in DNA replication.